Good morning. Today I'm going to be discussing the FDA's recommendations and recent updates for orally inhaled nasal drug products, also known as OINDPs. So the first part of my talk is going to give a brief overview of the orally inhaled nasal drug products, as well as some of the challenges that generic applicants have to meet when developing these products. I'll then go into the bioequivalence recommendations that we recommend and then finish up with some recent updates to these PSGs. So when developing locally acting generic drug products for OINDPs, generic applicants have to address many different types of challenges. The first set of challenges that I wanted to detail here are the patient-related challenges. So So these types of products are often indicated for the treatment of diseases that impact the respiratory tract, and these can include asthma, COPD, and rhinitis. And for these drug products to work, they have to reach the local site of action within the lung and nasal cavity. So it's important for their performance to, their performances will be dependent on factors such as achieving an adequate regional drug distribution at the local site of action. Another important point I want to note here for these products is when the therapeutic effect happens relative to when the drug is measurable in the blood. So for locally acting orally inhaled nasal drug products, these products have a therapeutic effect that which you can measure with a clinical or pharmacodynamic measurement that occurs before the drug can be detected within the blood. So your pharmacokinetic bioequivalence studies, which are often used for establishing equivalence for products that are systemically active, this type of bioequivalence study may not be sufficient for establishing bioequivalence for these locally acting products. Now, if we move to the device-related challenges, the first point I'll make is that orally inhaled nasal drug products are complex drug-device combination products. And so again, what that means is that in addition to the drug components and how that impacts performance, it's also important for generic applicants to consider the characteristics of the device and how those can impact drug delivery. Now, the device design can vary considerably across both the inhaled and nasal drug products. And to illustrate the scope of complexity with the types of device designs, I want to just give you a brief selection of some arbitrarily selected devices for orally inhaled nasal products. So starting with the orally inhaled products, you can have devices that are self-contained and multi-dose. You can also have devices that require the patient to open and load the dose before it can be delivered. You can have devices that require some input from the patient, such as pressing on a canister to initiate drug delivery, or you can also have some devices where the patient merely just has to inhale and that will trigger the actuation. Similarly, for nasal products, you can have multi-dose devices, some that are for single use, and you can have some that are for that need some type of input from the patient, such as pressing on a canister, or conversely, you can have some that the patient actually has to blow into a mouthpiece to initiate the drug delivery. So what I hope to illustrate here with these arbitrarily shown devices is, again, there's a wide range of complexity in terms of the device design, and so an important consideration generic applicants have to make is paying attention to the patient-device interaction, such as between the test and reference product, what's the user interface, are these comparable or substitutable, as well as is the patient using the device correctly. Now, if we move to formulation-related challenges, I'm going to walk you through a table that will show you a brief overview of the types of formulations we have for these products that are currently on the market. Of course, we have products that are both for inhalation and nasal route of administration, and both can have systemic and local sites of action. In terms of For the inhalation and nasal products, you can also have solutions, you can have suspensions, you can have aqueous sprays, you can have metered aerosols, as well as solid blend powders and even semi-solid formulations. So you can see, again, there's a wide range of complexity with the formulations that we have for these products, and each of these formulations will have their own different types of consideration and challenges that applicants have to address when developing these. And this can include the physical chemical properties of the API and the inactive ingredients, how these interact with each other, as well as how they can impact the performance of the product. And in some cases, for some of these products, whether the types and amounts of the inactive ingredients need to be controlled for when the applicant's developing their generic or in establishing bioequivalence. So in consideration of these challenges and the incomplete understanding of the relevance of results from bioequivalence studies to the drug concentrations at the local site of action, as well as uncertainties regarding the sufficiency of correlation of in vitro tests to in vivo PK data for establishing bioequivalence, we recommend the weight of evidence approach for establishing bioequivalence for locally acting orally inhaled nasal drug products. Now, with this approach, there are 4 components that need to be completed to satisfy the criteria. The first set would be the in vitro studies, followed by PK studies, compared to clinical endpoint or pharmacodynamic studies, and followed up with formulation and device sameness. Again, our product-specific guidances currently recommend this approach for locally acting dry powder inhalers, metered-dose inhalers, and nasal suspension products. This is just a table that shows the types of product-specific guidances we have for these products. You can see there's a wide range of active ingredients that we've addressed, and currently we have 56% of approved orally inhaled nasal drug products having a PSG. So again, you can refer to our product-specific guidance website to download these for your reference. Now, having described the weight of evidence approach, I'd like to go into some of the more details for the in vitro and in vivo studies. For in vitro bioequivalence studies, for DPIs, we recommend the single actuation content as well as the aerodynamic particle size distribution. I include other details here, but for the sake of time, I won't go into more detail. Metered-dose inhalers include the same types of studies, but also add spray pattern, plume geometry, and priming and repriming if that's required by the reference product. Similarly, nasal suspensions include these types of studies, but in place of aerodynamic particle size, we have droplet size distribution by laser diffraction, as well as drug and small particles or droplets. Now, for these in vitro studies, again, they need to be conducted on all strengths of the product with at least 3 batches of test and reference with no fewer than 10 units from each batch. And for metered-dose inhalers and nasal suspensions, the priming and repriming studies are recommended, again, if this is required by the reference product. So for instance, for breath-actuated metered-dose inhalers that don't require priming and repriming for function, we wouldn't recommend the in vitro bioequivalence study for priming or repriming. Now, if we move to the first set of in vivo studies, these would be the comparative pharmacokinetic studies. So for study design, we recommend a fasting single-dose two-way crossover with the objective of determining the difference in systemic exposure between the test and reference product. For these studies, the strengths, we recommend all strengths be tested since the relationship between PK dose proportionality across these strengths, as well as in vitro performance and product characteristics, is not well understood. In terms of dose, for the DPIs and MDIs, we recommend a minimum number of inhalations sufficient for characterization of PK using a sensitive analytical method. For study population, we recommend healthy subjects, and I include our conventional bioequivalence criteria here for your reference, but I won't go into more detail for these. Next is the in vivo bioequivalence studies for comparative clinical endpoint study design, as well as pharmacodynamic study, rather. So again, so these are for DPIs and MDIs, and they follow this study design of randomized, placebo-controlled, parallel, or crossover. So there's a placebo run-in period for DPIs and MDIs, as well as the placebo being needed for study sensitivity. So nasal suspensions are somewhat similar. Again, they recommend the comparative clinical endpoint study using the randomized placebo-controlled parallel group study design, and include the similar recommendations as the DPIs and MDIs. For the PK studies, again, the objective of those studies was for determining difference in systemic exposure. For these studies, this is for determining the difference in the local delivery at a site of action between the test and reference product. In terms of strengths, we recommend the lowest labeled dose, particularly for the comparative clinical endpoint study. For the dose, in terms of if it's single or multiple, this ultimately depends on the mechanism of action. So for your product, if it is fast acting like a bronchodilator, we'd recommend a single dose study. However, if your product is something that takes a lot longer for an effect to be seen, such as like an inhaled corticosteroid, we'd recommend the multi-dose study design. Nasal suspensions, we recommend multi-dose study designs, and this is mainly because these types of products often include an inhaled corticosteroid. So for study population, we recommend one patient population indicated in the approved labeling. And again, I include the bioequivalence endpoint criteria here for your reference, but don't go into more detail for the sake of time. The last set of products that I'll detail here are our nasal solutions. So our recommendations are ultimately dependent on the site of action as well as the type of formulation that we have for the product. So if your nasal solution is a systemically acting product and it has a Q1, Q2 formulation, we recommend in vitro bioequivalence studies that follow the same type and design as nasal suspensions, with the exception that there's the potential for in vitro study waivers for some of the studies for if there's if your product has a lower strength. Now, if your formulation is not Q1, Q2 the same, we recommend the in vivo PK study. Again, similar study design as the nasal suspension, with the exception that we recommend the highest dose be tested with the potential for a waiver of the lower strength. We don't have any non-aqueous based formulations for systemic activity for a nasal solution, so we don't currently have recommendations for these. We can move to locally acting nasal solutions. Again, these, if your formulations are Q1, Q2 the same, we recommend the in vitro bioequivalence studies following the same type and design as for nasal suspensions. And if for a non-aqueous based formulation that's locally acting, again, if your formulation is Q1, Q2 the same, recommend in vitro bioequivalence studies as well as in the same type of study design as for nasal suspensions, with the added note that these need to be conducted for all strains of the product. So after covering the recommendations, I'd like to go over just a few recent updates and notable additions to some of our PSGs, starting with the nasal suspensions. So as I detailed earlier, nasal suspensions follow the weight of evidence approach for establishing bioequivalence, and this can include a comparative clinical endpoint study. So for these products, the comparative clinical endpoint study is necessary because of an inability to adequately characterize the drug particle size distribution using conventional analytical techniques. Additionally, the drug particle size distribution in suspensions has the potential to influence the rate and extent of drug availability at the site of action in the nasal cavity as well as in systemic circulation. So with that said, there's been, of course, more advancements in analytical techniques that are capable of characterizing the drug particle size distribution, even in the presence of other suspended particles. Because of that, our locally acting nasal suspension PSGs now recommend new methods such as morphology-directed Raman spectroscopy, for providing comparative particle size distribution data in lieu of conducting the comparative clinical endpoint study. Now, again, this recommendations included in our nasal suspension PSG for fluticasone propionate, as well as for these active ingredients here. Lastly, I'd like to turn to our recent product-specific guidance for beclomethasone dipropionate metered aerosol for inhalation. So as I described earlier, the MDIs, they follow a comparative clinical endpoint study for establishing bioequivalence because they follow the weight of evidence approach as well. However, I'll note that the drug substance, that in the approved label for this product, the drug substance beclomethasone dipropionate is dissolved in the formulation. So in other words, this is a solution-based formulation. So with a solution-based formulation, and if a generic applicant has a test formulation that's Q and Q2 the same, and if the test device is sufficiently similar to the reference device in terms of critical design attributes and user interface, then there's the potential to use additional supportive data for establishing equivalence between a test and reference product for the local site of action and in the lungs. And so these additional studies can be used as an alternative to conducting the comparative clinical endpoint study. In terms of what these studies are, the PSG lays out some examples of what can be provided. I won't go into detail for these here, but if an applicant has wants to use this approach, we strongly recommend that they contact us through the pre-ANDA meeting process, because this helps us to clarify our expectations with the applicant in terms of how they're conducting their development program, if they have questions on how to do these types of studies, as well as for us to assist in making sure their ANDA is as completed as possible when they go through their submission. So in conclusion, I want to reemphasize that orally inhaled nasal drug products are complex drug-device combination products that have many challenges that generic applicants have to address. To help address these challenges, we publish product-specific guidances that can facilitate generic drug product availability and assist the generic pharmaceutical industry in identifying the most accurate, sensitive, and reproducible approach available, as well as identifying our current thinking methodology to support ANDA submission. So with that, I'd like to thank those here for the help in putting these slides together, and again, thank you for your attention.